And finally, our last section for physical principles is the cell membrane. We're going to be talking about the lipid bilayers and which ones are going to have the hydrophobic and hydrophilic portions. We're going to talk about different types of proteins that we find both in the integral of those known as transmembrane or the peripheral membrane proteins. And finally, we talk about the different types of molecules that can pass through very easily, which ones need help, which ones will need different types of proteins for either transport either into or out of the cell. And here we see the phospholipid bilayer, which you've seen quite a few times before. So we have both an inner and also an outer leaflet. So one will often face the interstitial fluid or oftentimes another adjacent cell, and one will be actually facing the cytoplasm. So remember, we have a polar head group, which is going to have a phosphate, as well as some other functional groups on top of them, whereas the hydrophobic tails will be more towards the inner and facing the inside of the bilayer. Now, each one of these individual phospholipids can fully rotate on its own, as you can see in blue, but also they can shift in what was known as a fluid mosaic model, where it can stay on its own side of the phospholipid bilayer, and very, very rarely, less than 1%, it may flip from one side of the bilayer to the other. So like I said, it's very rare. So if you can imagine just a bunch of balloons on a string, of course, millions and millions of these particular balloons, you can see all of the phospholipids moving very freely from one area to the other. So for some cells, this creates a very dynamic surface, whereas sometimes most of the proteins within those particular areas are often more stationary, especially if they're anchoring. But as far as some other proteins that may be receptors and such, they can move back and forth along this particular phospholipid bilayer. Lipids also tend to form about 50% of the actual mass of the bilayer, whereas the rest made up approximately of protein. The mitochondria, on the other hand, can be a little more on the protein heavy side because of the interactions of all the components for the electron transport chain, but for the most part, for most cells and other organelles, you will see the 50-50 split between the proteins and the lipids as far as mass for the phospholipid bilayer. Now, aside from the different types of membranes and different types of phospholipids we find in the plasma membrane, other things we may find will include glycolipids and glycoproteins. These can be used for either a slippery surface to allow certain cells to slide upon one another, or to act as tags. For instance, for red blood cells, often have a glycoprotein tag to show whether they are type A, type B, or both, or in the absence of all, be type O. But the other major factor, macromolecule that we see in the plasma membrane is cholesterol. Cholesterol is necessary to maintain a certain amount of fluidity found in the plasma membrane, especially in the colder temperatures. So whether it be for organisms like us or whether it be for plants, it prevents the solidification of the plasma membrane to allow certain things to pass through, whether through the bilayer itself or through certain types of proteins. So too little cholesterol and the plasma membrane can basically fall apart. Too much cholesterol and actually starts to solidify the plasma membrane. Therefore, no exchange can happen across the actual membrane itself. Here's a pic of the phospholipid bilayer or the plasma membrane with different types of proteins embedded within and sometimes through the plasma membrane. The ones you see here that will pass from the outside of the cell to the inside of the cell through the plasma membrane are known as integral or transmembrane proteins. If you notice here, the alpha helices that are found within these integral proteins are noted to be hydrophobic, so they tend to line themselves up with the hydrophobic area of the plasma membrane. This helps maintain the orientation of the particular proteins within the plasma membrane. So like begets like in this case, whereas the hydrophilic areas will tend to layer through either the outside cell, oftentimes to the interstitial fluid, or to the adjacent cell, or sometimes facing the cytosol. Oftentimes these will be also anchored by glycolipids on the outer portion of the cell, and sometimes by different types of fatty acids or prenyl groups or other peripheral membrane proteins on the cytosol side. Generally, these proteins tend to be either anchoring proteins or they tend to be tags as far as location tags or possibly even receptors for different types of either hormones or different types of neurotransmitters. The other types of proteins we see here are known as peripheral membrane proteins. These can either anchor the transmembrane proteins or form different types of enzymatic reactions. So in this case, the enzymes that we find with the electron transport chain will be under the peripheral membrane protein type, whereas other portions of that particular chain will be known as the transmembrane protein type. But we see a myriad of different types of proteins all throughout the cell, depending on the specialization of that particular cell. We contribute, thankfully, the fluid mosaic model to two scientists by the name of S.J. Singer and Garth L. Nicholson. Dr. Singer on the left and Dr. Nicholson on the right. Both of them have postulated that following the rules of thermodynamics and looking at different hydrophobic and hydrophilic interactions, they determined that the fluid mosaic model was the most apt model that we have for the surface of the plasma membrane, where different types of proteins as well as the phospholipids can move freely along their particular portions of the plasma membrane. Whereas before, most scientists have postulated that the plasma membranes were simply a combination of one layer of proteins with a sandwich layer of phospholipids in the middle, and also an inner layer of proteins. So one layer of proteins towards the interstitial fluid, the plasma membrane, and then the innermost proteins facing the cytosol. What they had done very simply is they'd done different types of stains to look at the different types of components of the cell. And of course, staining for different types of hydrophilic proteins, were able to see that they were freely movable all throughout the area. So they're able to use electron microscopy to also stain and be able to see the different components of the particular cell. So they're able to see the integral proteins embedded within the phospholipid bilayer, and this is one of the mosaic models that they posed through here with the globular proteins. Now, oftentimes we do see the transmembrane proteins or the integral proteins. We'll often have the alpha helices, which are going to be in the hydrophobic regions, deeply embedded within both sections of the plasma membrane. But they also may have different types of structures known as the beta barrel. So the beta barrel oftentimes will be used for different types of channels to allow certain things to pass through. So in this case, the types of molecules that we see pass through need some assistance tend to be your charged ions. So oftentimes they'll pass through the beta barrel. So you often find these in different areas of bacteria, mitochondria, as well as chloroplasts. So these types of proteins are known to be amphipathic, which means they are both have sections of water-loving areas and water-hating areas. So in this case, hydrophilic and hydrophobic sections. Now these will also anchor depending on which side of the cell that we're talking about here. In the outside of the cell, they will too be anchored by glycolipids, by what's known as the carboxy terminus, again found on the outside portion of the cell, whereas the inner portion of the cell are going to be anchored by other proteins and also by 14 carbon fatty acids by the amino terminus, which you might have seen quite a few years back or semesters back in Biology 1. Oftentimes it will also be attached by the palmitic acid, which is a 16 carbon fatty acid, and often with other types of what's known as prenyl groups with cysteine residues. So these are anchored not only with the hydrophobic groups found within the plasma membrane, but also by both the different termini, the carboxyl terminus, the C terminus, as well as the amino terminus found on the other end of this whole polypeptide chain. So again, we do see both the amino terminus and also the carboxyl terminus. Even at these large proteins, they'll have endpoints on both parts of the polypeptide chain. So we talked in this section before about the purpose of the plasma membrane is to maintain the cell's internal environment versus the outside environment. And it does this by allowing certain types of structures to pass through very easily and allowing other structures to either enter or leave the cell with a little bit of help. So very simply, the molecules that can pass directly through the plasma membrane without much interference include the steroid hormones like estrogen, which are hydrophobic and tend to be nonpolar. Other small uncharged molecules include your gases like oxygen and carbon dioxide, as well as nitrogen, and also water as well. Glycerol, ethanol, which are going to be your alcohols, can also pass through relatively easily. However, the larger molecules, especially the polar molecules, and even the ions cannot pass through very easily and need to go through certain types of channels, where a lot of times need to be consumed either phagocytically or through penocytosis. So the larger molecules, as well as the charged molecules or ions, tend to have to pass through proteins, and we see two different types. We see channel proteins, which again we talk about the beta barrel. This allows a nice, safe environment for the ion channels, the ions themselves, to pass through. So it maintains a hydrophilic tunnel through the actual plasma membrane. So we see on the left here, we see an ion passing through. So there's no impedance here, there's no gate at all. This was known as a porous channel or a leakage channel. We oftentimes will see this in some neurons as well as muscular cells in animal organisms. In the middle here, we see what's known as a carrier protein. These are going to be known as a passive transport, which very simply going from a high concentration to a low concentration. And this can be either from the outside of the cell into the cytosol or vice versa, depending on the concentration gradient. So of course, this does not need any ATP or energy drive to move these molecules back and forth. As long as there's a gradient, as long as there is enough carrier proteins available, that we can get the particular solute from one area to the next. So in this case, under the letter B here, we see the carrier protein will basically bind with this particular molecule. It'll cause a conformational change. So very simply, it will shunt the actual molecule into the cell from what it looks like here, and then revert back to its original form, waiting for the next binding site to be bound by another molecule here. So again, these are all passive transport requiring no energy. Inversely, if we're looking to drive different types of ions or molecules against the concentration gradient, we need to use a carrier protein and also use ATP to drive this particular shunting of these particular ions against the concentration gradient. So in this case, this is a proton pump. We see the proton will bind to this area of the carrier protein with the addition of the ATP donating its phosphate and phosphorylation of this particular carrier protein. Will actually shunt the proton into the cell at this point, and then water will come in and actually shunt away the phosphate or cause conformational change. This carrier protein will actually revert back to its original state, ready to be bound again when the next proton comes into play. Now, at the end of each chapter, they try and tie in some of the concepts in the last couple of chapters with some real-life applications. So very simple, what we're looking at here is a couple of HeLa lines of breast cancer cells. And what they're doing very simply is using the different types of data analysis, interactive testing, to basically look at different structures found within the cell. So if you remember, we talked about fluorescence with different types of targets of the particular types of the cell. So as you can see on the left here in blue, we have two different lines we're looking at. One is untreated breast cancer cells, and one is treated with 10 nanometers of paclitaxel. Paclitaxel is known as a taurine, which is basically a drug that helps suppress certain types of genes that basically will help promote cellular mitosis by trying to prevent the accumulation of microtubules for the mitotic division. By also targeting the gene, the FOXO3A. So very simple, what they're looking at is under the first section here, under the DAPI, what structures can you ascertain would that be? You kind of have to go into the merged section over here on the right-hand side to get an idea what those are. Obviously, the ones in blue are going to be the nucleus. Now, that's important because when you're looking at whether they're actually treated with this drug or not, so we're trying to see where the proteins will be dispersed into the actual cell. So that's going to be in the green fluorescent sphere. So if we're looking at the different FOXO3A gene, which is responsive for the production of microtubules, very simply, they're all over the cell, pretty much all around the nucleus, right to the periphery of the plasma membrane. And if you remember something about actin, actin is one of those proteins that helps form the cytoskeleton of the cell, one of the microfilaments there. So you can see under the merged section on the first row on the right-hand side, the blue would be the nuclei, the FOXO3A gene, or termed protein, is going to be all throughout the cell. So you can see it's pretty much widespread, and then of course surrounded by the actin. Now the nuclei, they're going to be treated by the paclitaxel. You can see that the protein stays within the actual nuclei, so therefore it's not being expressed. So the main thing with the FOXO3A gene is this basically is for cell program depth or apoptosis. So this is one of the target genes that some scientists are trying to target to stop the spread of certain types of cancers. So this is basically chemotherapy. So in this case, the FOXO3A gene, and therefore its protein that it codes for, does not ever leave the nucleus, so it stays right in a particular spot. So you look at the merged view, you can see very simply that very little of the FOXO3A gene has been expressed out into the cytoplasm. So if you want to go through the actual assessment at the end of the chapter, feel free to do so. I feel like the experiment does not go into too much detail about what the actual drug does, what it codes for, but this is more an exercise to give you an idea of the different structures found within the cell and how they differentiate from one section of the cell to the other. In this case, the nucleus, cytoplasm, and of course the edge of the plasma membrane for each individual cell. Just gives you a nice little synopsis of what kind of things that some of the scientists out there are looking for, how they're coding for certain types of proteins, and how they're able to track it and see if things will spread or it can be halted based on different treatments. Otherwise, you're all set to take the diagnosis quiz for Chapter 2. Again, remember you have 2 chances to submit, and I'll take the higher of the 2 scores. Good luck, and I'll see you in Chapter 3.